Hello, everyone. Thank you so much for this space to share with you, to have the opportunity to share with you our research that is today entitled Preliminary Insights from Population Genomics of the Weevil Metamasius emitters, assessing the genetic background for erythrocolor color. My name is Iván Fernando Calixto. I am a researcher from the Foundation of Science STAR that is based in Colombia. Why we are studying Metamasius hemipterus? Well, this is a huge problem in the department where we are based, that is called Casanare. In this province, there are several tropical crops that are quite important, like sugarcane, banana, plantain, and palm trees that are particularly affected by this insect. Its larvae feeds on plant inert tissues, causing necrosis galleries, and we in CESAR have already have the opportunity to use an approach that is metabolic coding, and we detect the Bursophilicus cocophilus that is a nematode related with the damage in these crops. Also, from a more like a biological perspective, it is quite interesting, this group, because it has a high degree of polymorphisms, uh, in which, due to the elytra color pattern, three subspecies can be differentiated: the Metamasius hemipterus carbonarius, the Metamasius hemipterus, and hemipterus sericius. The electrocolor is a multifunctional trait that likely integrates not just the natural selective pressures like camouflage or thermoregulation, but also sexual selective pressure like recognition and mate choice, and also potential protective physiological effects. For that reasons, our question in this project was: is there an association between electrocolor patterns in Metamasius hemipterus and their genetic background? And to address this question, we made an important broad sampling through the foothills of the eastern Andes mountain range in the province of Casanare in Colombia. We use pheromone to attract the insects in bucket type traps from palm oil, and banana crops. For the taxonomic determination, our taxonomics in CSTAR, we're able to differentiate several species, including Metamasius hemipterus, and for these species, they were able to group the samples, the individuals, in 23 different clusters of elytra color. So in order to get a high throughput sampling of variation across the genome of the individuals, we use a genotyping by sequencing methodology that led us to get thousands of SNPs markers homogeneously distributed across all the genomes of these individuals. This methodology recovers neutral loci to ask for questions related to population genomics, demography, or phylogeography, but also this kind of methodology detects outliers from the background that can tell us about adaptive processes. So for the DNA extraction, we use the QIAGEN DNA Splut and Tissue Kit. We perform some quality inspection like spectroscopy by nanodrop equipment to check for contaminants. And also we check for degradation by electrophoresis, because it's important to get a high weight molecular DNA. For the genome screen services, we made a contract with a company that is SNPsaurus. It's based in Oregon, United States. They offer us that for a small genome size like the one that we're expecting, that we are expecting something in this species like 800 megabases, to perform a new services that they have, that is a whole genome sequences for each of the 95 individuals, but at a low coverage, around 10x. It means to read each nucleotide of the genome at an average of 10 times. In that order, We send the nucleic acids they made, and they made the genome libraries and sequencing in an Illumina platform. One individual is then selected to assembly like a kind of draft reference to detect the variants and frequencies, producing a .VCF format that is the source of our analysis. Thanks to this sequencing effort, at the beginning, we were able to obtain more than 86,000 of variants for each of the 95 individuals. Then we applied some population genomics filtering steps like missingness per individual and missingness per loci, minor allele frequencies, Hardy-Weinberg equilibrium, linkage disequilibrium, and homologous elimination. So at the end, we were able to recover more 81 individuals and more than 13,000 of those size. Now, as results, we try to define the genetic cluster. So we applied several tests like the Bayesian information criterion and alpha scores. All these methodologies tells the same story, that the best case, the best number of genetic groups, it's one. It means any genetic differentiation is not enough to separate them in two or more groups, but it seems like a more uniform landscape in these genomes. Some explorative analysis checking for the 23 categorical groups of electrocolor that are you can see here at the right part. Like this PCA do not exhibit association, as you can see. And we will expect cluster of the same color and no more random distribution of the over the PCs. Even more, using a discriminant analysis that maximize the weight of our a priori of the electrocolor, the results are basically the same, with the ellipses overlapping at the space of the graph. Admixture analysis to estimate ancestry proportion at a K of 18, that is the number of sample sites where these individuals came from, and plotting them in a structure bar plot in the x-axis from the southern part to the northern part of the department of Casanare suggests a more genetic division into three main groups that correlate with their geographic origin, similar to a clan, with the south, center, and north, as can see more clearly when we plotted using K number of three. Overall, K number tests, principal component, and mixture plots preliminary are exposing no correlation with the electrocolor in Metamasius hemipterus, but their genetic background diverges by a more classical distant pattern, this having into account the natural history of Metamasius of extent of distribution. To finish, we had the opportunity to include some individuals from Metamasius hemipterus, one of the subspecies that has been proposed to be another monophyletic species, nested with Metamasius hemipterus carboniferous. Three individuals survived our filters and are signaled here in the graph in green arrow. Following this hypothesis, we will have expected them to have a similar ancestry profile and be close, but as you can see here, they do not have a similar pattern of admixture. And also they are not clustered near each other in PCAs or DAPCs. So at the end, it could be interesting to go deep into this data and to add other three more individuals that we already have, but that they were filtered out in at the beginning in the bioinformatics steps. Also, we want to perform non-supervised geometric morphometrics because we have already landmarks for each of these individuals that we're sequenced. Also, we are planning to do some analysis to detect outliers across the genomes that could be interesting to understand particular selective pressures at a genic scale. We hope at a bit more like a long-term project to understand what is the real background of the electrocolor, addressing this by other layers of information of the knowledge, like gene expression profiles or trying to get information from the epigenome. That's all. Thank you. We want to thank you all of you for your time, and we want to acknowledge the funding from Ministerio de Ciencias de Colombia, to Andrés Valenzuela, Elena Moya, and Jeanette Carvajal, that are collaborators in this project, and the rest of the staff of CESAR. Thank you so much.